I would like to kind of home in a little bit more into this question with you and Kate. Beyond thinking about how do we get to the right cell type, what do you think is the single most pressing question, right? We as a space need to address, and that can be scientific or clinical or manufacturing oriented, whatever you want to dig into here, that you have, right, about developing LNPs for specific next gen therapeutic applications, right? Gene editing, personalized cancer vaccines, etc. And we can start with Ben here, since you're the biggest one on my screen right now, and Kate.
No worries. It's a huge question, and it's almost impossible to say the biggest, because there's a bunch of different challenges, and so it is a challenge.
Top three, top five,
top three. So I think what would be brilliant is if, as a field, we could choose the winners better. So from the start, what is a winner and what isn't? What is going to give a better outcome for the patient when we're using that as a therapeutic? And that is a real challenge, and it touches on all of that complexity. So if you're developing, no offense to people that develop small molecule pharmaceutical drugs, you've essentially got one component and then you've formulated it into a tablet, a pill, or whatever. When we're doing the LNPs, as we all know, it's a lot more complex. And so what if you tweak something at the start that then would allow you to just seamlessly get through into the patient without those effects? How do we do that? There's so many aspects to that question, and so many ways we could approach trying to solve that as we move forwards as a ecosystem, and at all levels, the pharmaceutical companies, the academics, the medical research institutes. And so I think that is a big question. I think that's probably the most burning one. And the second question is what we've already touched on, is just for a particular target product profile, what is the best way to formulate that system with all of the variables we've talked about, the pH and composition and the mode of delivery, and so that's a challenge for the field.
Kate, what do you think? Are you in line here? Do you have other pressing considerations or concerns that you think, as we're looking at some of these next gen approaches for therapeutics development, that we need to be asking, or investments that need to be made, right, to get us on the right path?
Yeah, I think a lot of learning in the last couple of years. And I think especially when you talk about extrahepatic targeting, I think it's almost come to the point in my mind, instead of doing what we used to do in drug development, of drug design, in vitro, small animal, large animal, and then get to human, we may have to be more innovative here. Really coming in, understand the organ, the target organ, and then choose the right animal model. And may not be a small mice. You probably have to go in for rat at the very beginning. And then from there, trickle back into in vitro to do your large quantity screening. Because, again, with this protein corona, which very much impact on the biologic system we're tested in, and also the competition across different organs, and also the circulation, right? So those are the things make fundamental differences of where you can drive your a particle to. So, I've seen people spend tons of time doing in vitro, tons of time doing mice, only find out didn't go anywhere in the large animal model. So I think that's where, I'm just looking at the question 11 as well, and combined with what your question and Ben's comments is, how can we cut the chase? How can we be more effective screening the right lipid for the intended organ deliver the interested cargo. So those are the things need to come in together, asking the right question first, and establish the testing mechanism, and then go from there.
Okay. So, I mean, to ask a hard question, right? How do we cut to the chase? Are there investments we need to be making, or is it a matter of coming up with better models? I know, Ben, I have a question for you on this later.
No worries.
And we will jump into that, but I'm curious, Kate, to continue, and then Sumit, I want to talk to you.
I'm sitting with Sanofi BioNTech with all the money in the world. They can afford to make mistakes.
Pressure, guys, pressure.
They can send 100 people testing. So I think a small company needs to be really innovative. That's why, even though I have not seen much of it, 2 years ago, if you asked me, I'd say, AI is in the future. But now you force the company to think about how can we do faster? How can I accelerate this learning, and I still can be at a table with Sanofi about BioNTech, right? So I think those are the challenge we're thinking and struggle every day.
Comes back to the money.
To answer that question.
Comes back to the money, guys.
And I think you're thinking of some other company.
Yeah. Sumit, I want to home in here a little bit too, and then I do actually want to transition to some of the conversations about translation as well, and thinking about preclinical models. But I want to home in on CMC here too, because again, as we're looking at these next gen approaches, gene editing, personalized medicine, cell therapies, et cetera, right? Obviously, these raise a number of different questions and concerns than creating large-scale global vaccines, right? So I'm curious from a CMC standpoint, where do you think we're going to have some of the biggest challenges in the future here, and how are you sort of approaching what investments need to be made, right, to ensure that the process that you're building, the platform that you're building is applicable to these very different types of approaches?
That's a great question, and I'm actually incredibly lucky. In one sense, I focus a lot on vaccines. That's become almost accidentally my calling for the last 10 or so years. But I do work very closely. That's a story for another day.
We're gonna want that story later. Just saying.
Sure. But I do work very closely with my colleagues at Sanofi, who are equally passionate about using mRNA-LNPs in the therapeutic space, right? And we are trying very hard, in terms of at least infrastructure and manufacturing platform to keep things as consistent as possible. But you can imagine there are challenges, right? Of course, one of the biggest thing is the route is different, right? You go from intramuscular to intravenous, and that brings with it its own challenges. The dose range, the frequency can be quite different, right? So even in the early clinical stages, the demand can be quite different. So how do you design a facility which can, cover a spectrum rather than be, very rigid in terms of batch sizes and all, right? Speaking more about the route of administration, it brings along with it other challenges, essentially, right? Safety-related attributes, whether it's particles or endotoxins or double-stranded RNA, they're all important in both spaces, but the relative importance can be very different, right? So we have been trying to learn, try to better understand what is the impact of each of these depending on the route of administration, and how do we, control for them better. In future, I can also see that it may be beyond intravenous administration, right? So if you need a pulmonary delivery or something else beyond intravenous, now you're talking about device, right? So fortunately in pharma, many of us are well versed with the, developing combination products. You gotta keep in mind the constraints of developing a device along with the drug, essentially, right? So these are just some of the examples. I have probably 10 other examples, but I don't want to make specifically about this essentially, but you get the idea that, there are several implications on CMC as we are thinking of expanding beyond vaccines into the therapeutic space.